So we're going to talk about this week's experiment. And as you know, we have been working with E. coli. And this is an E. coli cell. And within the E. coli cell is a circular genome. And on that genome is a set of genes that we know really well, the lac operon. So the idea for this experiment is to put pieces of the lac operon into a plasmid. So here is the lac operon, here's a plasmid, and the idea is to get that plasmid into the cell. Alright, so let's talk about the lac operon for a second. Let's just remember what the lac operon looks like. And remember that there are three genes, LAC-Z, LAC-Y, LAC-A, and that those genes are all controlled by the promoter, which has three elements in it. This is cap binding. It's the promoter, where RNA polymerase sits down. And this is the operator. And if you remember, the operator is where the LAC-I sits. Okay. So the experiment that we would like to perform is to take this promoter and add it to something in which we can measure in a different way other than in the enzymatic beta-galactosidase. And so we've taken the gene that encodes green fluorescent protein and we've hooked it to the lac operon promoter with cap binding, the promoter, and the operator all intact. And to that, we put the entire thing into a plasmid. So in addition to having the GFP hooked to the entire promoter of the lac operon, we also have the GFP hooked to a truncated piece in which the cap binding site is missing. So we call this plasmid, P for plasmid, lac GFP. And we call this plasmid P minus cap, lac GFP. So what we're going to do is have E. coli cells that have their genome, and on their genome is the native lac operon. So these cells will be able to respond normally to the presence of lactose. And then we've made our plasmids that have lac GFP. And so these two E. coli cells, if this E. coli cell expresses the plasmid that has P lac GFP, And this E. coli cell has the P minus cap lac GFP. And we can ask a very simple question, and that is how does GFP get expressed in the presence of lactose or glucose or both? And how does that GFP expression compare to beta-gal? So remember, beta-gal is going to be coming from the lac operon on the genome, whereas GFP is going to be coming from the plasmid. Both are controlled by the lac operon promoter, but the output is different. And now, because the only difference between this cell and this cell is the presence or absence of cap, we can ask a very simple question. What is the contribution of cap to the expression of the lac operon by using GFP as the proxy? All right, good luck.